and he's a consultant pathologist and director of hematology flow cytometry at the American Hospital in Dubai. He's the inspector and fellow of the College of American Pathologists, Deputy International Commissioner. And the most important things about Aaron, if I speak of him personally, knowing him, he's the most inspiring person when you want to listen to a humble professor talking about how to transform science into practice. Listen to Aaron. Please, Aaron, if the floor is yours, and he will be talking about what laboratory manager needs in the era of Corona and artificial intelligence. Please, the floor is yours.
Masa here, thank you for the kind introductions and greetings from Dubai. And thank you for the invitation, Dr. Samia, Dr. Ali, the organizing committee. And I think I will focus on COVID and AI, and a lot of the themes will overlap with the first two speakers about the value of the laboratory. And so if I can get the slide deck, I don't know if it's showing up yet. So the objectives for today, we'll look at the brief overview of COVID-19, what our experience has been in bringing new tests to the lab in a pandemic, challenges for the next phase, and some of the questions we are getting in the lab. And then the fourth objective is positioning the lab for continued success. So these are the brief outline. And as you know, I guess we're getting close to the 1-year point in this pandemic. And I think many of us hope that this would be a short race, maybe a 100-meter dash, but it's probably more like a marathon. There's been a lot of information and data. The genetics of the virus was mapped and sequenced and published very early on, and I think that contributed to the laboratory's ability to respond. And now there's promise of a vaccine that's not only safe, but there's good evidence of efficacy and protection against the virus. So we're entering a promising next phase, although I think most of us hoped that there would not be a next phase, or it would have been an early knockout if we were in a UFC type of situation. So typically, and these are things that we already heard in some of the preceding talks by Dr. Graham, the lab quality, typically when we think about key performance indicators, we think about the different phases, about how fast we can turn around a test, about validating and verifying tests, and also understanding what the test performance is in the laboratory and application to the clinical setting as well. So one of our favorite stores in the UAE, and I believe this is also present in Saudi, is IKEA. And if you go to IKEA, that means you're trying to build something. And when you, for example, if you were to buy a stool or a table, you hope that it comes with instructions. Well, typically lab tests, we have time to look at instructions. The science has been looked at from many different angles and developed over years of research. In a pandemic, we don't have that luxury. And in many cases, it would be like going to IKEA, buying a piece of furniture, and coming to your home and realize there's no instruction in the kit. So you have to build this tool. And having a framework of lab quality helps you even when you have little data. And we're going to go through the example I will use is COVID serology, because I think we're still in the middle of answering what does that test mean. So typically we know the test performance characteristics, how it's going to behave in different phases and the impact on results. We usually have EQA schemes or PT testing. We have developed calibrators and QC, and the process is well mapped out before we have to bring a test to the lab and clinically. But this year, many of us are using lab tests that are under special marking. And you're very familiar with this marking of Emergency Use Authorization, or FDA EUA. Well, unfortunately for some, the EUA in some test kits actually had to be rescinded in some cases. So it's not a perfect process by any means, and the lab has to be very careful that when you put a test out and start offering to clinicians, you've done the proper studies and understand that not every test that comes to your lab is ready for releasing and for clinical use. So the type of tests that we've been using, predominantly molecular testing, real-time PCR, and in probably recent months, increasing understanding of the role of serology. And obviously the graph, which will come up in a later slide, shows the typical serological response, clinical response, and virological response. And this is worked out for COVID. But I usually teach the hepatitis session for medical students. And for hepatitis, the curves are based on long time of data points and understanding of when the clinical, the virological, and the serological response and the timeline of that occurs. And early on in the pandemic, I remember in February and March, people were publishing curves like this. And then over time, we realized, well, the curve actually is not quite what it starts to evolve over time as we get more data points. So it's always important to understand not only does prevalence in your community influence your test, but the actual curves and where you are in a patient's clinical setting, that will influence your test performance. And we've seen that quite a bit. So we still talk of the PCR as the gold standard. I think over time we realize that this gold standard is qualified. It is a laboratory or analytical gold standard, but it may not be a clinical gold standard. So some of the limitations are listed in the slide. I won't go through this, but obviously there's been examples in which patients are PCR positive or they PCR relapse, but it's not live virus. So it's a very sensitive test, but it's not perfect by any means when you think about lab tests and clinical implication. The other thing that lab managers have had to deal with is choosing the lab platforms and also dealing with uninterrupted supply chain. So these are logistical issues that we've learned to have to deal with in the pandemic. It's challenged our possibly traditional way of looking at starting lab testing and what are some of the parameters for choosing, either in terms of pricing, in terms of the ease of use of the platform, obviously access to ongoing reagents, and also having the test platform that will operate in your setting and that you can get it into your lab rapidly. Those are key considerations. And as I am involved in the CAP program, I would say that the college has done an amazing job in also supplying the regional labs in the GCC with proficiency testing and getting the testing kits and the programs to the labs in very timely fashion. And also in many of the cases, for example, in our setting in the UAE, the public hospital has done a great job because early on when the central labs were the only ones doing PCR, they set aside specimens in order to basically have a validation test kit for testing of PCR. So basically an internal EQA type of scheme. Because having EQA is very important because it gives us an opportunity to benchmark our performance, and that's very critical. So most of you who are part of CAP labs understand the importance of the test method validation and verification scheme. I won't go through this, but just to point out that under the FDA EUA and the CAP understanding that these would be treated as if they were FDA approved, and therefore the verification of these tests is required, but not a full clinical validation of the test. So obviously understanding the types of kits you have and then what steps you have to go through to document performance. So let's apply some of these principles and address what we had to deal with in the setting of serology. So two of the types, obviously the rapid lateral flows and then the non-rapid larger scale platforms that I think will see increasing use in the coming months. And some of the gold standard type of ways in which you can compare serology, obviously having serum of immunocompetent or patients that recovered, having correlation to viral assays. So most facilities will not have access to viral cytotoxic assays and protective assays. And then some people use plasma therapy as an indication of immunocompetent serology or a way to interpret your serological response. So this is just pointing out the Mayo Clinic early results on recovering serum and their correlation with COVID serology in their setting. So again, use of serology, the types of test is important. The timing of when you do the test is also important. And this is just a summary of some of the ways to look at a validation set. If you're planning, if you haven't validated serology, I think most labs have already validated some of the automated serological tests. So ideally you have a gold standard kit, or you have titers and quantitative tests, although qualitative tests is seeing a significant role. So you need to have examples of positive samples from patients, and typically in the early validation sets, they were patients that either cleared the virus and or showed clinical improvement. There are some examples of positive cases in which we saw a small percentage in documented PCR negative patients. Those were interesting in the early months of the pandemic. And it's important also to have negative samples in this case as well. And the PCR of low-risk cases of other coronaviruses, as well as HIV, and avoid comorbid or autoimmune patients as well. So this is from a CAP Today article, and the article, I would refer you to it. It was about in September, October, that talked about the steps to verifying SARS-CoV-2 antibody assays. And obviously precision and comparison studies are important. And the timing of when you do your serological testing is key. Early on in the pandemic, I would be discussing with many of the labs that were looking at bringing on serological studies. And when I asked them what they were testing at the time of PCR positivity. And they were wondering why the test performance was less than optimal. And as it stands, most serological tests are not as sensitive as PCR. But if you test before day 14, and oftentimes even before 3 weeks, you're not looking at the peak serological response. So the timing of your serological testing is going to be important in how your validation test and your validation and verification study performs. So some of the uses, confirming classic cases that may be PCR negative. We've seen a small percentage of cases, classic COVID clinically, PCR negative. Epidemiological studies for seroprevalence, the documentation of immunity, and obviously we'll need to have functional correlates. And then as vaccines become deployed, the efficacy of vaccine, and I think we'll be seeing a lot of data points over the next probably 6 months until next summer or fall on correlative studies of vaccine, its efficacy, and serological studies. So nowadays, as we go into the final months of this season, one of the obvious questions that we often get asked, and is very relevant, and when we look at the partners that are bringing on diagnostics, people have been planning and being preemptive about this. And that has to do with the overlap of the upper respiratory viral or flu and RSV season with COVID. We knew there was a high possibility that this was going to happen. And this is our data from actually the first 4 months of this year, where we were doing a syndromic panel. At this time, COVID was not integrated in the panel. So we were doing a 2-step testing algorithm. And basically what we found was that the co-infection rate was fairly low, and that by and large, if you found a respiratory viral pathogen, the likelihood that that patient had COVID was fairly low. But again, you need to validate it in your setting. But going forward in the next probably 4 to 5 months, how you approach COVID in respiratory viral season and flu season is going to be key. That's going to be a question that's going to be asked by your pediatricians, by your respiratory clinic, by your emergency department clinic, as well as many anxious patients and parents. So is it flu? Is it COVID-19? Is it RSV? Because they're going to be approached and treated differently. Now, this is an algorithm that Mayo Clinic has developed, and you may want to study it in more detail, but different institutions, different settings are going to approach it differently. And obviously having access to rapid testing, ruling out flu, ruling out RSV, ruling out or ruling in COVID-19 will be key as we enter the next couple months of this year. If you have the access to NGS and sequencing and can provide you important data, we had some early results from the medical school as well as the Al Jalila Children's Hospital, one of the main molecular and NGS labs, that showed the sequence and phylogeny of the virus, and it's very important for epidemiology to show the relatedness and where your viral strains are coming from. I think over time that will probably give us additional correlative information as we have more information, and clinical symptoms as well as possible vaccine efficacy may relate to the actual viral sequence, and that's going to be interesting data in the coming months as we have more information. And obviously we're still learning about the long-term effects of COVID. And there are many different organ systems that we're observing that they're long-term, for example, cardiac effects, neurological effects, as well as other end organs. And the pathology community will obviously contribute much to the understanding of the long-term effects. And these are areas in which we still need reliable markers to know what is actually a long-term or chronic effect of COVID-19 as we go forward. So in summary, it's ideal to have multiple tests, the molecular, the serological ones, and there's some places obviously looking at antigen testing in the algorithm, other diagnostic tests that are important, and this will relate to your diagnostic setting. Are you in a reference lab? Are you in an emergency setting, possibly a physician clinic or a point of care setting? So CBC, NLR, SED rate, chest x-ray findings of ground glass opacity, temperature, and O2 set. So all of these are data that will feed into large data sets that are useful in not only providing diagnostic information, but also triaging and clinical information for treatment. So the value of the lab in a novel pandemic, we talked about some of these aspects, quality and timeliness of results. Obviously, as we are able to bring reliable tests to the lab, address the logistical issues. A lot of these are around pre-analytic registration, post-analytic reporting, and tracking. Know the questions that are needed and what will be addressed in the coming months, and know the limitation of your test, what's the sensitivity and specificity, and also how it relates to your specific community and the time and the patient. And always have a multi-pronged approach to how you provide testing, especially in this setting. So I think I've lost one of the slides, but I will end with the summary on this. We have a lot of data that's coming in for COVID. There's a lot of volume of data. There's a variety of clinical data as well, and we have to validate or prove the veracity of the data. So these days, when we think about AI, we need to know what kind of AI platform are we dealing with? Are we working within an existing EHR? Are we working in the context of a health information exchange, which has really large data platforms and access? And then the training design of your software is very important, and then validating your AI software. And generally, there are three pieces that I think are low-lying fruit as I approach AI projects these days. One of the low-lying fruit is automating processes that are fairly manual but are repetitive. So AI can contribute in the automation of repetitive processes. The second one has to do with leveling the playing field. So AI can make a generalist have access to analyzing data that typically a specialist physician or even a super specialist would perform. So AI can often level the playing field and give power a lot more to practitioners in rural areas and also primary care gatekeepers. And then the third one is what I call the brute force needle in a haystack. So those are typically the three types of AI projects that we're looking at. Automation, the provision of super specialty integration of data sets so that the gatekeepers has way to triage data. And then the third one is kind of defining the hard to find data in a large data set. So that's just a very quick overview, and thank you very much for your attention.
Thank you, Dr. Han. This is you are putting us back on the track as in respect to the time. And at the end, there will be answers and questions at the end of the session. I know it's strange to speak over the virtual. Always the networking and the live conferences make the sessions more attractive, more energetic. You will see the reaction of the audience. But this is one of the outcome of the COVID-19, and hopefully we would be optimistic that the networking, the informal, even Dr. Graham said that the virtual, or you would like to see as well back the interaction and informal networking. It is my pleasure to introduce the third speaker, Dr. Hisham Shams. Dr. Hisham Shams is a very well-known speaker.